Okay, welcome. This is going to be the section on genetics. I'm speaking right now to an empty room, so this is an SDL version of this lecture. And the first thing we're going to talk about is mutations and polymorphism. So you'll notice we have a section here on genetics. I think there's something like 13, 14 lectures. The first 5 lectures I'm going to do, and then next week Dr. Gardner is going to come in and do the rest of the genetics section. I sat in on his lectures last year. He's awesome. I really enjoyed them, and I think you will too. He's been doing this for a long time as well. And he's a real specialist in genetics. So I'm going to cover the fundamentals at the beginning, and then he's going to come in and do some of the more, I guess, advanced topics, if you will. But I'm going to cover the fundamental stuff first. And the first thing we're going to start with is the mutations and polymorphisms. Okay. You'll notice I haven't put some objectives up yet. I will get them up very soon. If you don't see those very soon, send me an email and say, hey, where are the objectives? And I'll make sure I get those posted. But before we actually start diving into looking at some of these mutation types, now you might notice the first few slides we're going to go through hopefully they look a little bit familiar to you because we went over this a little bit in the section on translation, and we're going to go over it again to make sure you understand. And in order to understand some of the mutations that we're going to talk about, certainly you need to be able to understand the genetic code. And when you change nucleotides, how that can alter ultimately what becomes a protein. Sometimes changing the nucleotide doesn't change anything, but sometimes it does. And that's our very first types of mutations that we're going to talk about. So, the first mutations we're going to talk about are single nucleotide changes or point mutations. Now, before we get into looking at some actual point mutations, there's 2 different versions of this point mutation. You can either have a transition or a transversion. So a transition is where you have a purine that's replaced by a purine. For example, an A replaced by a G, or a G replaced by an A, or a pyrimidine replaced by a pyrimidine. C replaced by T, T replaced by C, something like that. Or you can get a transversion, and that's where you go from a purine to a pyrimidine, or a pyrimidine to a purine. So in this example I've given you going from an A to a T, or from a C to a G. And hopefully if you remember earlier when we talked about nucleotides, which ones are purines and pyrimidines, which you still need to know, then you would be able to recognize when you happen to see a mutation of that type, whether it's a transition or a transversion. So yeah, you still need to remember which ones are purines and which ones are pyrimidines. If you remember, I gave you that little thing to help you remember pure as gold, that little mnemonic, or cut the pie if you prefer that one. Either way, if you know one, then you automatically know the others. Okay, so if we mutate this DNA sequence, we have several possible outcomes of these single nucleotide changes. We can have a silent mutation. So this is where we've changed a nucleotide, but when we go back and look at the genetic code, it still encodes the same amino acid. Okay, and we've given you an example here, UCA. If you look up in your code, that codes, or in your genetic table, that codes for serine. If we change one of the nucleotides, if we change that A to a U, okay, then what ends up happening? Well, if we look in the genetic code, then it still encodes the same thing. It still encodes serine. So that's what's called a silent mutation. In most cases, a silent mutation is not going to be of any genetic benefit or detriment. So it's going to be genetically neutral. That's not 100% true because some organisms have a codon bias. They prefer certain codons, but for the most part, that's true. Next we have our missense mutation. So this is where you have a codon encoding a particular amino acid. You change one of the nucleotides, and now it encodes a different amino acid. Now there was a mistake in your PowerPoint. I've corrected it on this version that I'm using right here, so you can write it in. I still have the UCU here instead of the CCA. So an example of a missense mutation would be UCA. We're starting with the same codon we did last time and mutating to CCA. So we mutated that first nucleotide, and now when you look it up in the genetic table, you'll find out that that encodes proline. So we went from a serine to a proline, which is actually a pretty significant change. You guys already know a little bit about the amino acids and what they look like. Certainly you didn't have to memorize the structures, but you know what is basically you know that a serine looks like. You know it's got a hydroxyl group certainly, and you remember proline had a very specific structure. So that's a fairly significant change. We might guess that that may actually have some significant effect on the protein. Now, part of the reason I start thinking in those terms comes with this additional type of mutation that is a missense mutation, but we use this term conservative mutation if you have a missense mutation that results in an amino acid with similar properties. So if you change from a nonpolar amino acid to another nonpolar amino acid, in particular if the R group is pretty close to the same length, probably not going to be a significant change. It could be, but in many cases it won't be, and that's an example of a conservative mutation. And certainly when you look at human genes, you can find all different alleles, all different versions of a normal gene. When you start looking at some of those, many of these differences are conservative mutations. They don't change the function of the protein, therefore they're genetically neutral, and therefore they're not selected against. And in certain populations, certain alleles will be more popular. So that's an example of a conservative mutation, something you may run across. It's still a missense mutation. So generally speaking, the more drastic the amino acid change, the more you would predict that it's going to have an effect on the protein. That's not always 100% true, but it's a good guess before you know anything else. A real-world example, and something I believe you've already seen, is sickle cell anemia. There's a good example of a missense mutation, good example of a single point mutation. The normal codon is GAG, and the mutant codon is GTG. Or if you're using the genetic code that I gave you, GUG. Okay, so the U substitutes for T in RNA, right? And when you actually look those up in the table, you see it goes from a glutamate or glutamic acid to a valine, which is a pretty significant change. You're going from something that's negatively charged to something that's completely nonpolar. And I think you've already went over the mechanism of how that ultimately affects the protein, and then how you actually form those fibrils in the red blood cells that ultimately make them misshapen and cause the hemolytic anemia and the occlusive crises that you see in sickle cell anemia. Okay, so sickle cell anemia is a very good example of a missense mutation. The next type of mutation is a nonsense mutation. So we just talked about a missense mutation, now it's a nonsense mutation. And this is where you go from an amino acid encoding codon to a stop codon. And I've given you an example here. UCA to UAA. So we went from that C to an A, and now we went from something that encoded serine to a stop codon. Okay, and that's a nonsense mutation. And as I mentioned to you guys before, the ones that people tend to forget or confuse are, because silent mutation is pretty obvious. A lot of the other ones we're going to talk about are pretty obvious. The missense and the nonsense are the ones that usually give people problems. So I just always say, remember something with non-stop in it. I said here molecular biology is non-stop fun, which I think we can all agree with. So, or you can put whatever you want in there. Dr. Lucia is non-stop fun. Whatever you want to put in there, but non-stop. Just remember, nonsense mutation is a stop codon. Okay? And that'll help you remember. So once again, that's the one that always tricks people. A real-world example of where you will see nonsense mutations in a disease we've already discussed in several different lectures, and I believe you will actually discuss in next week with Dr. Gardner a little bit in some of the advanced topics on nonsense mutation in PKU. Okay. So just to remind you, most cases of PKU are caused by a mutation in the phenylalanine hydroxylase gene. There are other versions of PKU, which if we haven't already talked about, we will talk about. But right now we're talking about, in particular, the mutation in the phenylalanine hydroxylase gene. This gene actually converts phenylalanine to tyrosine, so it's involved in the catabolism of phenylalanine, the breakdown of phenylalanine. And we went over this a little bit, and how you can actually treat the disease by restricting the diet of phenylalanine. Now with PKU, there's over 400 known mutations. Actually, I believe there's over 500 known mutations. Anyway, there's many known mutations. And here's kind of a cool figure. It's a picture of what the phenylalanine hydroxylase gene looks like, and it's drawn relatively to scale. And this is really the first time we've, I think anyway, looked at a human gene in this class in terms of how it looks on a chromosome. And these little red slivers in there, those are actually the exons, which get joined together. And these sort of light yellow parts are actually the introns. So one thing that immediately, at least jumps out to me, is the fact that the introns are enormous compared to the exons. And that's very typical for a human gene. Human genes, the intron regions tend to be absolutely enormous. So you'll see genes occupying just enormous sections of a chromosome, when actually the final messenger RNA, when all those exons are spliced together correctly, is a much tinier part of that. So that's kind of neat to see, and kind of a little bit bewildering as well. Now, because there's all these different mutations, now right now we were talking about nonsense mutations, and you certainly do see nonsense mutations in PKU. It's one of the types of mutations you can see. There are also missense mutations. You also see splice site mutations. We'll talk a little bit about splice site mutations a little bit later, and we'll also talk about insertion and deletion mutations later. So, as I go through this section, you'll notice that I'm going to talk about a number of different diseases. Some diseases, like sickle cell, for example, you had a very specific type of mutation caused a very specific disease. And I would like you to know that one, okay, because that's a high-yield, real-world example. You also get conditions like PKU where there's tons of different mutations, okay? It's a good example of a nonsense mutation, which is where we're using it, but I think it's also important to know that there's a whole bunch of different mutations, because that ultimately affects some of the tests you might try to do. Because you're not looking for a specific mutation here, it kind of limits some of the genetic tests that you can do. We'll talk about that a little bit later. So beyond the point mutations, we have other mutation kinds. So first, we're going to look at our insertions or deletions. And I believe we've also went over this before, but so let's just go over it again. So you can have, we categorize the insertions and deletions based on whether or not they're a multiple of 3. Okay? So a multiple of 3 would be 3, 6, 9, 12. Either insertion or deletion of that many nucleotides. Okay, so we're talking about nucleotides here. If it's not a multiple of 3, meaning you have a 1 nucleotide insertion, 2 nucleotide insertion, 4 nucleotide insertion, something like that, we're going to have what's called a frame shift mutation. Why do we call it a frame shift mutation? Well, if you remember way back when we did translation, that very first start codon, that very first AUG defined the rest of the open reading frame. Everything was broken up from there into triplets. Okay? If we insert or delete some nucleotides and it's not a multiple of 3, everything beyond that insertion now, you've actually changed the reading frame. That means everything downstream of that now encodes different amino acids. So it becomes a, downstream of where the insertion or deletion is, becomes a completely different protein. And you can either take my word for it, or you can take a sequence. You can do this on the computer. You can go into GenBank. There's all kinds of programs. You can pull up a sequence and insert something in there, and you'll see it changes all the downstream sequence. Usually, also what happens is when you shift a reading frame, an open reading frame, you usually introduce an early stop codon too. So you change the open reading frame to a completely different protein, and usually you have an early stop codon as well. So you end up truncating the protein too. So if you looked at an individual and you did a Western blot, for example, from them for this particular protein, you would also notice that it would be shorter than normal in most of the cases. Another generality, the closer to the beginning of the protein, the more severe the mutation. The closer you get to the beginning, the more you're going to alter that protein. Sometimes if you get a insertion or deletion that's near the end of the protein, it might not be as severe, or it might be compatible with somewhat of a normal function, maybe reduced in absolute efficiency, but could still do something. But the earlier it happens, generally speaking, the earlier where the frame shift happens, the more severe the mutation. If the insertion or deletion is a multiple of 3, then it's called in-frame. So you either have frame shift or in-frame. So an in-frame insertion, let's say I insert 3 nucleotides. Basically what you do is you add an extra amino acid in there. But the downstream open reading frame is still in the same frame, so it still encodes the same amino acids. It's similar with the 3 nucleotide deletion. It doesn't change the downstream reading frame. So one real world and really good example of the difference between a frame shift mutation and an in-frame insertion or deletion, one of these indels, is Duchenne and Becker muscular dystrophy. Okay, Duchenne is the more, and we're going to talk about these, so you don't have to write this down right now, but when we get to the X-linked conditions, we're going to talk about these, but Duchenne is the more severe version of the disease. It also happens to be the one where you have the frame shift type mutations, where in Becker you either have an insertion or deletion, but it's a multiple of 3, so it's an in-frame, so it's not as severe. Both of these conditions are relatively severe conditions, but Becker is less severe, as we're going to see. So that's a real good example of the difference between an in-frame and a frame shift mutation. And we're going to talk about Duchenne and Becker muscular dystrophy a little bit later this week. Another example of an in-frame deletion, although this one happens to be severe, even though it's still in-frame and happens to be severe, is cystic fibrosis. Now, before I get into the details of this particular mutation, there are a bunch of different known mutations, and I forget how many. There's a lot of different known mutations that cause cystic fibrosis. The one we're going to talk about today is one of the most severe versions. Okay, so it's going to cause the most severe disease. There are mutations that are known that cause cystic fibrosis, but the symptoms aren't quite as severe. So you do get a range of symptoms in cystic fibrosis, from the most severe versions which we're going to talk about, to some less severe versions. But approximately 70% of the cases of cystic fibrosis worldwide are due to a 3 base pair deletion, okay? A very specific mutation in the coding region of the CFTR gene. This stands for cystic fibrosis transmembrane conductance regulator. It's much easier to say CFTR. And what this does is a chloride transport protein, so you have a defect in this chloride transport. Now we deleted 3 base pairs, so what's going to end up happening? Ultimately, you're going to be missing an amino acid from that protein. Okay? Downstream of that, still in frame, so all the rest of the amino acids are there. In this case, we're missing a phenylalanine. Phenylalanine is missing. You may run across, because this is a very famous mutation, you may run across this nomenclature, this CFTR delta. When you see delta in genetics, it means something is missing. Delta F508. So 508 happens to be the position of that particular amino acid, and F is the single letter abbreviation for phenylalanine. So that's a very famous mutation, and you may run across that nomenclature. So in some cases, you just delete a single amino acid. It's hard to predict exactly, but that doesn't sound too bad. But in this case, it does result in a very severe effect, because what it does is it doesn't allow the CFTR to fold correctly, okay? And when things don't fold correctly, and I believe we've talked about this already, but if I didn't emphasize it enough earlier, things don't fold correctly, the cell recognizes that, and it targets them for degradation. So you get improper folding and assembly of that CFTR protein, and it gets prematurely degraded. Now, cystic fibrosis is the most common lethal genetic mutation. Now, there are other more common genetic mutations in Caucasians, but this is the most common lethal mutation. I will also warn you that cystic fibrosis, I think we've already touched on it. I'm going to give you a little bit more about the pathology on the next slide, just because you need to start becoming familiar with it. This is a very high-yield disease for board exams. Okay, they expect you to know just about everything with cystic fibrosis. So I'm going to fill you in on a little bit of the story, but certainly you're going to expand upon it in other classes. The incidence is 1 in 2,500, which for a lethal genetic disease, that's incredibly high. It's incredibly high. So one of the questions you should be asking yourself is, why is that so common? And we're going to talk a little bit about that later on in the complications lecture, complications of some of these pedigrees and genetic complications. Okay, so the carrier rate is 1 in 25. We haven't done some calculations yet. We're going to do those probably on Wednesday, and how you will be able to tell me how to get this number from given an incidence. It's not hard, but that surprises people sometimes. 1 in 25 Caucasians is a carrier for cystic fibrosis. So that means that some of us in this room are carriers for cystic fibrosis, just by random chance. Actually, a significant number of us will be. Okay? So a little bit about cystic fibrosis, because once again, you got to kind of start grinding this in your head, start really becoming familiar with this disease. A clinical diagnostic hallmark, this is a test they use that's specific to cystic fibrosis, is elevated chloride levels in sweat. That's specific to cystic fibrosis. Remember, it's a chloride transport protein. They can measure that in the sweat. That typically, the chloride being in the sweat itself is not a pathological issue. But the lack of chloride secretion in the lungs becomes an issue. Okay? It leads to dehydration of the mucus. So you get mucus that gets very thick and accumulates, and it ends up obstructing airway passages. As a result, they're predisposed to recurrent lung infections, and usually that's what actually ends up killing a patient with cystic fibrosis. It's this pulmonary insufficiency, or a lung infection eventually will end up killing them. Now, historically, death often occurred in childhood, and patients rarely survive beyond the age of 30. We're getting better and better at treating some of the symptoms of cystic fibrosis, so that number is being moved, so patients are living longer. And certainly right now I'm talking about the most severe version of cystic fibrosis. There are versions that are not quite as severe, which could be compatible with a little bit longer lifespan. Another thing that's a hallmark of cystic fibrosis, in most patients you will also see pancreatic enzyme sufficiency, okay, and poor nutrition. That's because basically the same thing happens in the pancreatic duct. You get that real thick mucus accumulation blocking the pancreatic duct. You can't send those digestive enzymes out, so they don't digest food properly, and therefore they have poor nutrition. If you see somebody with cystic fibrosis, very often they look very frail and skinny because they can't get adequate nutrition as well as a person who doesn't have this particular mutation. They can also end up with, for example, because of that, these pancreatic enzymes, they're involved in breaking down fats and ultimately digesting. They can very often get deficiencies in fat-soluble vitamins. That's something that's very common in patients with cystic fibrosis, and something you have to be very aware of. Okay, so here's a nice little figure I just took out of one of the board review books that kind of shows you, I think, all of an example of all of the different mutation types that we've talked about so far. It doesn't have an in-frame insertion or deletion, but it's got a frame shift, showing you a frame shift. So that can help you just visualize what's going on. I just threw that in there for an FYI. Now, there are other types of mutations that can happen besides these single point mutations. For example, we can have splice site mutations. Okay, so these are changes in nucleotides involved in splicing RNA. We've already talked about splicing when we talked about transcription, right? That's joining those exons together, taking out the introns. Now, when we went over that, I told you you didn't have to memorize the specific sequence at those junctions, but I told you it was very sequence specific. So where the junctions between an exon and an intron are, there's very specific nucleotides. And that tells the splicing apparatus what needs to be spliced out and what needs to be joined together. If you have a mutation in those specific nucleotides, it can result in incorrect splicing, which can result in possibly deleting nucleotides from an exon and or leaving nucleotides from an intron in the messenger RNA. So that could cause an insertion or deletion ultimately in the final messenger RNA. You could end up completely deleting an exon, I shouldn't say completely deleting an exon from the final RNA, or potentially you could leave a complete intron in the RNA. It could be either one of those. Okay, so this will screw up splicing in one way or another. So remember, even though you don't have to memorize the sequence, there is a very specific sequence for the splice sites, and if you mutate that, you're going to cause incorrect splicing. The mutations that could cause that could be single nucleotide point mutations, could be insertions, deletions, things like that. But they can definitely be single nucleotide point mutations. And sometimes what ends up happening when that gets mutated is you'll sometimes produce a correct messenger RNA, but you'll produce a large population of incorrect messenger RNAs as well. So there's various levels of how this mutation will affect things. One real-world example of where you can see splice site mutations, and this is the one that's really very commonly noted in genetics textbooks when they talk about splice site mutations, is beta thalassemia. Since you guys aren't here right now, I can't ask you if you've seen the thalassemias yet, but I'm going to spend some time in the next couple lectures talking to you about thalassemias. So if you haven't seen them yet, I'm going to give you the basics of how they work. So, but just real quick, before we actually get to more in depth. So a thalassemia is a deficiency of either the alpha or beta chain of hemoglobin. Okay, if you're deficient in the alpha chain, it's an alpha thalassemia. If you're deficient in the beta chain, it's a beta thalassemia. Okay, so many of the common mutations in beta thalassemia are splice site mutations, and you can see that with this little dotted line here. And you'll notice there's a lot of different ones in there. Okay, splice site mutation. So you'll notice some of them are right in the middle of an intron. So what's going on there? That's not right at the junction. Well, you could create a new junction if you mutated a nucleotide, right? And then you'll end up thinking that this part of an exon is supposed to, or a part of an intron ends up in the final messenger RNA. So you can do all different kinds of mutations that will cause splicing defects. As a group, the alpha and beta thalassemias are the most common single gene disorders in humans. That's why we're going to spend some time talking about this. High yield diseases, something that's frequently covered in depth in genetics. We're going to talk a little bit more about those later. But splice site mutations do happen. Now, you'll notice there are some other mutations. One of the others we're going to talk about before the end of this lecture. So there are other mutation types, but splice site mutations are common in beta thalassemia. Another type of mutation you may see is a trinucleotide repeat expansion. Probably one of my favorite ones to talk about because these are so bizarre. Okay, so things you don't, when you think about genetics and think about how genetics works and how mutations work, this is not something that immediately jumps to mind. So these trinucleotide repeat expansions, now we're just going to talk about trinucleotide repeat expansions. It doesn't have to be a trinucleotide. There can be 2 nucleotides, there can be 4 nucleotides, but most of the repeat expansions that you see are trinucleotides. And this is a region where a sequence of bases, even in the normal person, is repeated many times. So you have, in the normal person, okay, before we even get to the mutation type, in the normal person, you have a sequence repeated over and over again, okay? But what happens in the pathological situation is this repeat gets expanded during DNA replication. And this repeat gets expanded significantly, okay? It gets amplified. How exactly does that happen? Well, we're still working on figuring that out. So somehow that repeated region messes with the polymerases and causes them to slip or something like that, and they'll expand that region. And you'll notice, and we'll talk about this later on in more detail, that as it gets passed down from generation, it tends to get bigger and bigger and bigger. Okay, so that repeat gets expanded even more. Another weird and interesting thing about the trinucleotide repeat expansions is, well, it's easy to imagine if you had a trinucleotide repeat expansion in the coding region of a protein, well, you're going to mess up that protein, right? It's trinucleotides, it's going to be a multiple of 3, so you're going to have extra of some particular amino acid. The N-terminal and the C-terminal region of the protein would be fine, but somewhere inside there, wherever that nucleotide expansion repeat occurred, you would have multiple of some particular amino acid. It's easy to visualize that can screw up the protein. But there are actually many examples also of where these repeat expansions actually occur in non-coding regions. So you can have these repeat expansions in the 5' or 3' untranslated region of a messenger RNA. Okay? Now that's a little bizarre. You might not think, okay, well, who cares? The coding region is still correct, right? Why would that mess it up? Well, there's a couple things that can happen. If you get a greatly expanded repeat in a untranslated region of a messenger RNA, the cell recognizes that there's something wrong with that messenger RNA. And sometimes these repeats can be thousands of base pairs. A normal untranslated region on a messenger RNA is relatively small, 30 nucleotides, 50 nucleotides, maybe 100 nucleotides, but it's somewhere in that area. Okay, and you know how translation works in eukaryotes. You scan. What I didn't explain in depth is the 3' and the 5' untranslated region interact, and they actually help translation. You get a huge untranslated region, the cell recognizes that, okay, something's wrong with that messenger RNA, and what ends up happening is it degrades it, and very often, because it recognizes that one is something's wrong with it, it'll go into the cell, into the nucleus, and methylate that particular gene and shut it off. Okay? So then that gene becomes no longer available. And I'll explain how that's going to affect symptoms. You can also, this aberrant mRNA can also be toxic. So you have, for example, sometimes these messenger RNAs don't get transported out of the nucleus, and they stay in the nucleus and they start binding proteins that are supposed to be involved in splicing, and they tie them up, and then you can't get correct splicing of other proteins, and so that mRNA becomes a poison, it becomes a toxin. Okay? And we have examples of both of those situations. Okay, so we're going to look at a trinucleotide repeat expansion in each of those different regions. The first one is in the 5' non-coding region. The most famous example is Fragile X syndrome. Okay, so Fragile X syndrome is caused by an expanded 5' untranslated region. So you have a trinucleotide repeat, and I didn't even give you what the specific repeat is. It doesn't really matter, but it gets expanded greatly. And it's in this particular gene, FMR1. This is a very high-yield, famous genetic disease that you will be expected absolutely to know for your board. So any little details I give you about it, you should be familiar with, and put them in your long-term memory, please, because I want you to really be familiar with this disease as you move on. Okay, so this FMR1 gene, this is an example where that expanded repeat gets recognized by the cell as being, okay, this is a defective messenger RNA, let's turn this mRNA over, and let's go into the nucleus and hypermethylate that gene and shut it off. We don't want to make this messenger RNA, okay? So what is that going to do? Well, it's going to produce less protein, okay? Now, if you might think, okay, well, you've got 2 copies of each gene, so we got another copy of this, and if it's normal, then we should be okay, because this is a recessive, well, actually, I call it a recessive condition. Some people call it a dominant condition, and we're going to get to that a little bit later on. So there's a little bit of controversy, at least in my mind. Anyway, this actually happens to be on the X chromosome. So if you see this repeat in a male, they only have 1 copy of this gene, and if you shut it off, they're not going to produce this protein. Okay. Now the situation in females it becomes a little bit different. And I'm going to expand on that, and I think on Wednesday or Thursday, we'll talk a little bit more about that. The major clinical outcome from Fragile X syndrome is mental retardation. So this FMR1 protein is an RNA binding protein. So it's normally supposed to bind RNA, and so it affects cells and how they develop and how they're regulated. It's most abundant in the brain and the testes, and so those are going to be where you're going to see the major symptoms. So with the brain, you're going to have mental retardation. You have some very characteristic facial features. and here's a picture, and you can certainly type Fragile X into Google Images and find all kinds of pictures. This is a relatively common disease. Long face, large mandible, large everted ears. These are still all within the spectrum of normal. Okay, I sort of look like that kid in there. So they're all within sort of the spectrum of normal. And I don't have Fragile X, promise you that. But you still see this feature in all people who have the disease, okay? Or most of the people that have the disease, you see these characteristic facial features. The only distinctive feature that you see at least 90% of the patients in postpubertal males is this macroorganism, or very large testicles. Okay, so remember, FMR1 protein is most active in the testicles and in the brain. This is the most common inherited cause of mental impairment. Now, there's a more common genetic cause of mental impairment, which would be Down syndrome. But Down syndrome, you're not inheriting a mutation from your parents. Okay, they don't have Down syndrome, at least in most of the cases of Down syndrome. They don't have a mutation. This is something that occurs as non-disjunction, and we'll talk about that a little bit later on. But in this case, you actually inherit the mutation from a parent. Now, they could be asymptomatic, and we'll explain why a little bit later on, but you got, at least the pre-mutation was present in the parent. This is the most common known cause of autism. Now, it's a very tiny percentage of autism patients, probably like 1%. But in most cases, we don't know what causes autism. So, but the people with Fragile X will have autism-like symptoms. And I've given you a couple frequencies here. I don't expect you to try to memorize these, and they're not. Depending on what source you look at, you're going to get a different number. That's why I can't hold you to the number. Okay, and that goes with the other diseases that I'm going to talk to you about, because if you read a different reference, you might get a little number. In many cases of Fragile X syndrome, they might suspect it, but unless they do a genetic test, they can't say for 100% whether or not it's Fragile X or not. Most of the time, or maybe not most of the time, but in many cases, the parents might not be able to afford that type of testing, or maybe they don't even want to know to that level of what difference does it make, what caused the mental retardation to the parent. They want to know how to deal with the situation. So you'll get different numbers because of that. An example of a trinucleotide repeat expansion in a 3' non-coding region is myotonic dystrophy, or this is the most famous one. I'm giving you the most famous of the trinucleotide repeat expansions. There's actually more than what I'm going to talk about. Most cases of myotonic dystrophy are caused by a trinucleotide repeat expansion in the 3' untranslated region. The Fragile X is 5'. This time we're talking about the 3' untranslated region of the messenger RNA. This happens to be a protein kinase gene, and there's the name of the gene, DMPK. Okay, so usually with the gene names, they're italicized. Can you see that? And it's involved in muscle contraction. And what myotonia is, what you see in myotonic dystrophy, is a delay in the relaxation of the muscles. So you have a sustained contraction of the muscles, and that causes some of the characteristic features you will see on these patients. It also causes heart arrhythmias. Okay, so it's causes defects in both skeletal and heart muscle. And so at the molecular level, what's going on is this aberrant mRNA remains in the nucleus, and it binds RNA binding proteins involved in splicing. Right? This is an mRNA that's supposed to be spliced, but it doesn't get transported out of the nucleus. It remains in the nucleus, and it binds up those proteins that are supposed to be involved in splicing, including splicing other genes as well in these muscle cells, and then causes them to be defective. So you have a deficiency of this particular protein kinase that's involved in muscle contraction. It can also affect the expression of some of the other genes in those cells because of binding up some of these proteins involved in splicing. So it's a toxic mRNA. Okay, this is going to be an example of a gain of function mutation. I'll talk to you a little bit later on about what that means. This is the most common adult form of muscular dystrophy. When I say adult form, with myotonic dystrophy, at least in the first generations to show symptoms, you usually don't see symptoms until they're well into adulthood. That means as a child the symptoms aren't obvious. And we'll explain a little bit more about that later on. So there are more common form of muscular dystrophy. Duchenne muscular dystrophy is actually more common than this, but you see the symptoms in early childhood. So this is the most common adult form. So finally we have a trinucleotide repeat expansion, which is a little bit easier to explain how it works, with Huntington disease. And with Huntington disease, the repeat is in the coding region. So you end up having a repeat of a glutamine codon. Okay? This glutamine codon repeat causes the protein to be unstable and accumulates in aggregates. Okay? This instability actually takes a while. So Huntington disease is also another disease which actually has an adult onset. Okay, you typically don't see the symptoms in the first generations to show Huntington's disease, and I'll show you why it's important that we differentiate between the first generation and later generations. You might not see symptoms till they're in their 50s. It's important because at that point, if they were going to have children, they probably already had children, and we'll talk a little bit more about that later. Incidence about 1 in 10,000. So for a genetic disease, that's pretty common, and what ends up happening is this Huntington protein is actually most abundant in neurons. It precipitates, and when any situation where you got a protein precipitating in a cell and aggregating, you're going to cause that cell to die. So you'll see the same thing with Alzheimer's disease. Okay, for example, another example where you get a protein aggregating, precipitating, and then those cells end up undergoing death. And in this case, it happens to be nerve cells. So you get a progressive neurodegenerative disease. And I'm sure you're going to expand upon Huntington in some other classes. We're going to talk about it some more as well. Okay, you can also have, so let's move on to the next mutation type. You can have a large segment insertion or deletion. So in this case, I'm not talking about a few nucleotides. I'm talking about a big chunk of a chromosome missing. An excellent example of where you will see that. And so what happens there is you get aberrant recombination. So you get unequal crossing over during meiosis. So that's when you exchange gene fragments during meiosis. You get an unequal crossing over, so you end up with too many genes on one chromosome and not enough on another chromosome. Okay? A very well-known and famous example of that is alpha thalassemia. So I already mentioned beta thalassemia as a splice site defect. You can also get your alpha thalassemia. Alpha thalassemia, usually alpha thalassemia are caused by missing genes. Okay, very famous example of where you have a large-scale deletion. Okay, and I'm going to talk to you in the section on autosomal recessive diseases a lot about alpha and beta thalassemia. Okay, so I'll leave that story till then. Here's another table for you to use, to study from. It also gives you the effect on the proteins, which is kind of neat to look at. It's not, you can't always 100% predict what a mutation is going to do, but you can make some predictions. So there's also another type of mutation that could occur, which is a promoter mutation. We've already talked about promoters, what their function is. You can have mutations in a promoter region which alter gene expression. Most frequently when you get a promoter mutation, you could potentially have either one, but most frequently we think of these promoter mutations as causing a decrease in expression of the protein. One real real-world example of where that actually is known to occur is in some cases of beta thalassemia. We already talked about you can have splice site defects. We can actually have other defects as well. But you can also have a promoter defect. And when you get a promoter defect, you end up producing a normal protein, but you just don't produce as much of it. Okay, so that actually means, that actually complicates the clinical picture of the beta thalassemias. And we're going to talk about that in our section on autosomal recessive diseases. So you can get beta null versions where basically you get no functional protein from that particular copy of a gene. But you can also have these beta plus mutations. And a good example of a beta plus mutation where you produce a normal protein, but just not as much of it, is from promoter mutations that you'll see in some cases of beta thalassemia. Okay, finally, the last topic to talk about is this term polymorphisms. Okay, and I believe Dr. Gardner is going to expand on polymorphisms a little bit, but I want to define a polymorphism versus a mutation. Okay? As a very basic definition, before we even get into this, most of the time when people say mutation, they're thinking about something or they're talking about something that causes a disease. And most of the time when somebody says polymorphism, they're talking about some genetic change that ultimately doesn't affect fitness. It's genetically neutral, okay, so it doesn't either increase or decrease fitness, so there's no selective advantage or disadvantage, okay? That's kind of a general, so when you first see those terms, but it does get a little bit more specific than that. So if you look at two humans who are not related, about 1 in 1,500 nucleotides is going to be different. So everybody's going to have a whole bunch of differences in their nucleotides. Although everybody is fairly genetically homogeneous to a certain extent, depending on your perspective. So these genetic differences can be either called a mutation or a polymorphism. And sometimes that definition gets skewed a little bit. So only a few percent of the genome encodes exons that actually code for proteins. When you mutate those things, very often you have a functional significance to that mutation. Therefore, you're going to cause a selective advantage or disadvantage to having that particular allele. Right now we're thinking of diseases, so we're thinking of disease alleles. So most of the time we're thinking of a mutation causing something bad, but it could cause something good. But there's all types of intervening sequences that, as far as we know, if you change those, it has no consequence on the fitness or the gene expression of that individual. So those are genetically neutral, they're not harmful. Therefore, they tend to persist in the population. They're not selected against. So you'll find these to be very common. Okay? No selection pressure. These regions tend to be hypervariable. So if you look at individuals, you'll find lots of differences in those particular regions compared to actual gene regions. Polymorphisms may be so most of the time, those polymorphisms are in regions where you don't have exons and it doesn't affect gene expression, but they could be normal variants of a gene. You definitely have, if you start investigating genes and you looked at a particular gene amongst all of us, you very frequently will find changes in the genes which had no functional significance, okay, which didn't give an advantage or a disadvantage, and therefore they tend to persist, okay, so they're not selected against. And that would also be an example of a polymorphism. And usually we reserve the term mutation for genetic changes that cause a disease. Now there is a very strict definition of mutation versus polymorphism, okay? So a DNA sequence a sequence change that occurs with a frequency in a population of less than 1% is called, strictly speaking, a mutation. Whereas if it occurs at a frequency of 1% or more, then it's called a polymorphism. Okay, that's the strict definition, and you should be familiar with that definition. Okay, that 1% thing, because that's really, strictly speaking, what it is. But here's the issue. We have known disease genes that occur at a frequency of more than 1%. Okay, we looked at cystic fibrosis in Caucasians. You certainly see genes for the sickle cell mutation can definitely be at a higher percentage than 1%, especially in certain populations. So, and we would still call those mutations, but technically speaking, that's actually a polymorphism. Okay? So the definition can get a little skewed, and so you just have to be careful of that. Very often when we're talking about genetic tests, we talk about polymorphism. Sometimes the polymorphism is a disease-causing mutation as well. Okay? So if you're familiar with the strict definition, also be familiar with how those terms are generally used. Okay? All right, so that's all for today's lecture, and I will see you in the next SDL this afternoon.